And I hand over the chair to Gerson, my co-chair.
Okay. Well, the co-chair. Our next speaker is Dr. Purnananda Guptasarma. Sorry if I didn't pronounce it right. So he's from here, the institute in Mohali, India. And he talked to us about the intrinsic blue fluorescence in ordered and disordered proteins. Please.
Thank you, Gerson. My name is very unpronounceable anyway, so Indians have trouble with it. Thanks, Amit and Gerson, and thanks, Samrat, for the opportunity to speak at this meeting. I work on a variety of intrinsically ordered and intrinsically disordered proteins, but I'm not going to speak to you about any of those pieces of work today. Instead, I've chosen to speak about something called intrinsic blue fluorescence, which I hope many of you will find interesting as a tool with which to study intrinsically disordered proteins. And I'm going to actually tell you a story. Part of the reason is because after lunch, people are a little sleepy, and so I thought I would present this like a detective story to try and get you interested and wake you up. And the story is about the discovery and understanding of a new observation relevant both to ordered and disordered proteins, which happened in my lab around the turn of the century, a few years after that. And of course, with the years, it's become easier and easier to tell this story because the discovery itself has now gotten validated by evidence from over 10 research groups, including some which are represented in this meeting, such as the group of Dobson, Kaminsky, Udgaonkar, Samrat himself, Uversky, and others. And most of these groups have agreed with the explanations and the implications of this discovery. I haven't yet told you what the discovery is about, but I'm just trying to build up the interest and the mystery a little bit here. And the interest in the discovery and its applications appears to be growing. So last night I looked up the citations of two papers that we published, one in the year 2004 and the other in the year 2008. And this is the combined citation number. And you can see that it's been going up, and it's suddenly doubled to well over 20 in the year 2017, which is what partly also made me choose to speak about this in this meeting. And between 2004 and 2008, after we published the first paper, I almost didn't mention this paper by Shukla and others to anyone, because people would ask me why I was the only person in the world who had worked on this, and if what I was saying was true, why it wasn't in a better journal, and I didn't have any answer for that. Of course, in 2005, Daniel Odzen very kindly invited me to a European meeting on protein aggregation in Aalborg, and I did speak about this briefly in passing for 2 minutes out of a half-an-hour talk. And also Samir Maji, who spoke yesterday, he visited my group in another institute before I moved to IMTech, and in 2007, he and I briefly spoke about it. And then suddenly there's been this explosion of interest, and the reason I picked this is also because, if not all, a very large majority of the people who've been citing this report are from the field of intrinsically disordered proteins. So what was this discovery all about? It's about a hitherto unknown chromophore, which is also fluorescent, which turns out to be present in all proteins. And it has implications for a variety of areas, including molecular imaging, cell biology, spectroscopy, crystallography, protein aggregation studies, understanding of biological electron transfer, electronics, and diseases that occur in electrically conductive tissues, such as the heart or the brain. And, but let's start this at the very beginning. So you all know that ordinary proteins that lack prosthetic groups have 4 types of chemical groups which absorb electromagnetic radiation through electronic excitation. The first of these is, of course, the peptide bond, which has 2 kinds of transitions. And these are between 160, 170, and 210 nanometers deep in the ultraviolet. The second is the indole moiety in tryptophan. The third is the phenyl moiety in phenylalanine, and the fourth is the hydroxylated form of the phenyl moiety in tyrosine. And all of these of course, with very different molar extinction coefficients. This is a non-continuous, non-linear scale here. They all also absorb deep in the ultraviolet. So all 4 of these groups actually absorb either in the UVC or in the UVB region, which are defined here, with maxima of absorption below 310 nanometers. And the 3 aromatic groups, they fluoresce in the UVB or the UVA region, but not in the visible region, with the exception of tryptophan, which has a very mild residual fluorescence above 400 nanometers. As you can see here, that's 400 and 420 nanometers. So by about 410 or 420 nanometers, the fluorescence of tryptophan is gone. So before the year 2004, the canon in the field held that proteins neither absorb UVA light nor fluoresce visibly. Now in 2003, we were playing around with some designer proteins which we had labeled with fluorescent isothiocyanate and tetramethyl rhodamine isothiocyanate. And we were having fun mixing these colored proteins by co-aggregating them under common conditions to see whether we could create co-aggregates that were actually mixed at the molecular level and whether we could do molecular painting with these sorts of labeled proteins. And one of these was the backbone reverse form of a cold shock protein A. The other was another form of the same thing, just differentially labeled here. And we also did these experiments with another protein which was a backbone reverse form of a heat shock protein. And we were using confocal microscopy to see whether we could see fluorescent signatures of both labels within identical volumes in the sample. And we were asking questions essentially about whether molecular recognition is important in such aggregation or whether intermolecular cross beta sheet structure can cause the formation of what eventually turns into amyloids. And then we saw something entirely unexpected, and that was that in control experiments, we found that unlabeled protein aggregates display a strange blue radiation when they're illuminated by ultraviolet light from a mercury lamp. And so look at the center of this. This is basically a protein aggregate. Don't worry about what's on the sides right now. You illuminate this with light from a mercury lamp, and you see a strange blue radiation. And we saw this in amorphous aggregates that contained amyloid microstructure. So both with Congo red, which is shown here, and with other dyes, we showed that as these dyes end up getting taken up by these aggregates from the periphery inwards until the entire aggregate is covered, basically the blue fluorescence gets replaced by the fluorescence from the Congo red. We also saw this strange blue radiation in amyloid fiber bundles. These are some electron micrographs of backbone reversed CSPA shown here, and some visible light microscopy of fibers of diameter of about 0.1 microns shown here, where the same fibers, when they're illuminated with ultraviolet light, give you this strange blue radiation. And then we were wondering, what is this? And then we saw that in UV laser illuminated, not mercury lamp, but UV laser illuminated protein powders and various kinds of protein aggregates, amorphous aggregates, and as I shall show you later, also fibrillar aggregates, using a laser, which is the Coherent Enterprise 2653 laser, which has 2 lines, 351 and 364. We were able to show that gamma-2 crystalline-derived synthetic beta sheet-forming peptides show this radiation. Just gamma-2 crystalline whole, which is thermally aggregated, shows this. And we saw this also in a variety of different protein crystals. These are some crystals of P. furiosus, triphosphate isomerase, Hennig-White lysozyme, and various other things, including a hydrogenase and so on. We saw it in about 10 different crystals. And we also found that it is possible to distinguish protein crystals which show this radiation from salt crystals, which are also often present in hanging drops and sitting drops. So for instance, here you can see that if it's a salt crystal, then in the background of a protein aggregate which has just deposited out of solution, which is fluorescing brilliant blue, the crystal is dark. And if there are aggregates stuck on the side of such a crystal, you can see the fluorescence from those crystals. Whereas if the crystal is made of protein, then you actually see the whole crystal fluorescing. So naturally, we became curious about this strange blue radiation. And we wondered, is this non-fluorescent based? For instance, could it be a scattering artifact, such as either a Rayleigh scatter of some previously unknown component in the laser that nobody talks about, or could it be a Raman scatter of the Rayleigh scatter, involving the solvent inside the crystal or inside aggregates? Or could it be fluorescence-based, but not owing to anything in the polypeptide, for example, due to some cofactors? Or if we could rule that out, could it be due to some unknown properties of polypeptide components, for instance, some previously undiscovered characteristics of the aromatic amino acids, or could it be an entirely novel fluorescence? So we set about eliminating these different possibilities systematically. And the first was whether it's a second order Raman scatter, a Raman scatter of a Raman scatter of the 351 or 364 nanometer line, which gives you fluorescence at 440 or 450 nanometers. So we decided to excite at two different excitation wavelengths and see whether the emission wavelength peak changes. And if it doesn't, then it cannot be Raman scatter. And if it does, then it's probably Raman scatter. So forget about these panels for the time being and concentrate on these, which show you a lysozyme crystal excited with 351 nanometers and 364 nanometers. And we used a 11 nanometer bandpass spectrofluorimetric attachment connected to a confocal microscope in which we were doing the imaging to collect these emission envelopes. And you can see that the emission peaks, of which there are multiple, and which I'll have something to say about, they don't really change as you change the excitation wavelength. And also, if you look at the emission over a variety of emission ranges, you can see that the spectral band is too broad for it to be Raman scatter or any kind of scatter. And so the radiation seemed to have too large a bandwidth to be Raman scatter, and therefore it could not be a scattering artifact. Could it be a Rayleigh scattering artifact of something which is there in the laser that we don't know about, or could it be fluorescence? So we thought, let's approach this directly and try quenching the radiation with potassium iodide. And also, if you can see quenching, try photobleaching, and if the radiation disappears, then it must be fluorescence, and so try fluorescence recovery after photobleaching as well. And so this shows you a bunch of aggregates on a slide where from the side of the cover slip, potassium iodide has been allowed to come in. And as the potassium iodide comes into contact, you can see that this overexposed aggregate begins to show detail as a function of time. And then when you leave it for long enough, basically all the fluorescence is gone. So the fact that potassium iodide was quenching the fluorescence, quenching the radiation, established that it is fluorescence. And then we picked a region of interest, photobleached it, and then allowed some time to pass to see whether there was recovery of fluorescence after photobleaching. That was there too. And so all of these things clearly established that the radiation that we were seeing was some sort of fluorescence. And so we had eliminated this, and so we began to get interested in that, which is, could it be fluorescence owing to some non-polypeptide components? So we tried many different proteins, and we saw the fluorescence in all of them. And none of them had any known or isolated cofactors. We were still seeing the radiation, so we concluded that the cofactors cannot be responsible for the fluorescence. So we came to the third possibility, which is that it is fluorescence, but it owes to some unknown properties of known polypeptide components. So we prepared recombinant, so we were wondering about whether it could be photooxidative modifications of aromatic amino acids. So we prepared recombinant protein in the dark and asked whether you still see the radiation, and you did. And so it couldn't be photooxidative modification. And then we were wondering, maybe it's an entirely novel fluorescence, but we did one more experiment before we gave up on this third idea, and that I think is coming in one of the future slides. But at the end of all this, we thought when all else has been eliminated, then that which remains, however improbable, must be considered seriously. So at this point, the radiation had shown up in amorphous aggregates, amyloid aggregates, protein powders, protein crystals. We had not yet seen it in proteins in solution, but we saw it a little later. The radiation showed up in proteins sourced from vertebrate tissue, synthetic peptides, commercially sourced proteins, recombinant proteins, purified from different organisms and so on. And so we thought it must owe to some natural protein component. And there are only two chromophores in proteins that absorb ultraviolet light. As I said in the beginning, one is the peptide bond, the other is the aromatic amino acids, and nothing in proteins is known to emit visible blue light. So we checked sample aromatics to ask whether they still show the radiation. And this is one such sample. We've tested several. This is a synthetic peptide with a sequence drawn from gamma-2-crystalline, in which we had some other interest at that point. And this is the sequence. It's a 14-mer. You can see that there are no aromatic amino acids and that the radiation is still seen. So the radiation is not dependent on aromatics. And that left only one possibility open, which is peptide bonds. So we tested a dry powder of glutathione, which is a tripeptide which is gamma-glutamyl cystenyl glycine, and there was no discernible blue radiation, or at least the quantum yield of any fluorescence that was there was so much lower that it was as good as non-existent. So we saw a black field of view with no discernible fluorescence, which is why I'm not showing you that micrograph. But the problem was that, as already mentioned, the peptide bond itself was not known to have any fluorescence property either. And here we had a tripeptide which was not showing the fluorescence. So we were wondering what it is. And then in an unexpected flash of insight, a totally novel possibility suggested itself. And this is the possibility which suggested itself. It is that long wavelength excitation, because we're dealing here with 351 and 364 nanometer laser lines, very far away from the 282 nanometer that excites tryptophan. We're dealing with long wavelength excitation. So what about the possibility that delocalized electrons, in other words, lone pairs, and their quantum tunneling across hydrogen-bonded peptide bonds could be responsible? In other words, what we are talking about is not an n to pi star or a pi to pi star or a sigma to sigma star, but an n to a diffused fluor of some kind, a diffused orbital. Basically, the peptide bond has a partial double bonded character because there's a lone pair of electrons on this nitrogen and another lone pair on this oxygen. This lone pair on the nitrogen moves into the CN bond and exerts electron pressure and pushes the electrons here in this double bond, thus causing a lone pair here on the oxygen to transiently turn into an oxyanion. And then a little later that comes back and pushes back so that it goes back all the way to the nitrogen. And this resonance is what gives the peptide bond, which is a sigma bond, a partial double bonded character of 40%, which is what caused Ramchandran and Pauling to hold that the peptide bond is planar and unrotatable. And what we were wondering is whether instead of involving any of the electrons in this bond, which are classically responsible for peptide bond absorption, whether the electrons here, backed up by electrons coming from this nitrogen through this entire system, could be responsible. And our hypothesis was that these electrons, which are already quite delocalized from both the nitrogen and the oxygen nuclei, were getting further delocalized because of the hydrogen bonding that this oxygen is involved in, and that they were getting even further delocalized because the hydrogen bonding doesn't end here. You could have alternating networks of peptide bonds, peptide bonds, hydrogen bonds, peptide bonds, hydrogen bonds, giving rise to a new diffused chromophore. In other words, we're talking about these electrons not now returning back, but jumping across from one peptide bond to another peptide bond, and then through these bonds to another peptide bond, and through those bonds to another peptide bond, and so on, essentially kicking electrons down the road like you see in this cartoon, where each of these men is a peptide bond, and each of these kicks is a hydrogen bond, basically. And this electron, this can is the electron from the lone pair on the oxygen, right, from the carbonyl, basically, which is backed up by the lone pair on the nitrogen, with both of them arranging for electrons to slosh around inside proteins like a C in an alpha helix, where you have hydrogen bonds which are roughly parallel to the axis of the helix. The electrons would rush from peptide bond to hydrogen bond like this. Of course, in a beta sheet, they would rush across the sheet, across the strands, perpendicular to the strands. And these would potentially allow proteins to conduct electricity and could explain how rapid electron transfer happens either within proteins or between proteins in reactions that require instantaneous transfer. And this could also explain why there is this novel long wavelength absorption and fluorescence. And we thought, well, since it involves the peptide bond, as it did appear at that point, maybe it would show some secondary structure dependence and some conformation dependence, so it could be a really neat tool. Now I'm going to summarize this for you with a graphic, which is not, of course, a video. It would have been better as a video. I'm sure all of you who visited science museums have seen this thing, which is called a Newton's cradle, right, where you have these metal balls that are in touch with each other. And if you move one metal ball and let go, as the pendulum swings and it comes and strikes this ball, then this last ball leaves because the energy is transferred through this entire system. So what we are saying is that protein complexes are basically not insulators of electricity as was previously believed, but conductors of electricity. And these electrons from the nitrogen and the oxygen are jumping through these alternating networks of peptide bonds and hydrogen bonds throughout the protein. And so that is what enables an electron that comes in here to exert an electron pressure that pushes an electron out from somewhere else, very much like what happens in the energy transfer in this Newton's cradle. And indeed, one Professor Rama Sharma, now long retired from the Indian Institute of Science in Bangalore, got very excited by our papers. And he said, Purnanand, you know what? I've analyzed every crystal structure that's there in the database till now, and I've never found one which has any catalytic activity which lacks a helix. So I think that you should look very carefully at how helices in proteins are directed towards catalytic centers. And maybe this whole business of electrons rushing around and sloshing around could be used with helices acting, particularly because they have this helix dipole, as vacuum cleaners or hoovers, basically to suck electrons away from some part of the protein or blow electrons back into some part of the protein so that they're available for various kinds of reactions. And you can see I've just picked one random example from the net. So this has a folate bound here and an ADP bound here, and you can see that there is a helical bit of the protein which is directed this way. And so you can see this is a handheld vacuum cleaner, and I'm representing it as a helix, basically to say that when the electrons are rushing through all the hydrogen bonds going up the helix, maybe that is actually transferring electrons from one part of the protein to another part of the protein, allowing catalysis to happen. And so basically, this is the summary. The peptide bond normally absorbs at 200 to 250 nanometers. It has delocalized electrons. There's less energy for excitation because the electrons are less tightly held to the nuclei, and that explains why the excitation happens at longer wavelengths. And peptide electrons undergo two delocalizations, one through resonance, giving the peptide bond its double bonded character, and then further through hydrogen bonding, which was the novelty of this work. And we suggest that that's what was giving rise to absorption of either 351 or 364 nanometer light, in which case it could be a novel fluorophore, and explain why all proteins containing any kind of secondary structure should show it. Now, I must remark right at this point, in case I run out of time, that it's not just proteins that have secondary structure which show this fluorescence. In fact, right at the end, I'll talk about some work, very important work from the group of Uversky and Muschol. But in the meanwhile, let's look at the secondary structure dependence. These are different proteins. What you can see is that inside this envelope, very much the same bands are present in all the panels, but the relative intensities of the bands vary from protein to protein. And we see something at 425, 445, 465, and then a couple of things at further longer wavelengths. And you can see that the relative intensities of these vary quite a lot as you go from one protein to another. But the peaks are still discernibly at the same positions on the wavelength axis. And we think that this is a secondary structure dependent fluorescence from the peptide bond, which in the times to come might actually become a very useful tool for protein structure. Now, the question then was, if that is the case, because there is so much secondary structure inside even a monomer of a protein in solution, why shouldn't proteins in solution display these spectra? And so we went looking with tissue-derived gamma crystalline, as well as this previously described non-aromatic containing peptide, and we found that you do see this fluorescence. The only problem is that you have to use very high concentrations to see it. The reason you see it in aggregates and crystals is because in space the protein molecules are very concentrated, so that helps you to see it. Just like when you have a diffused fluor in a microscopic view, you just find a diffused background fluorescence. But if all of it gets titrated to one point in the view, then you see a bright fluorescence. So for the same reason, it's useful to see this in solids, but you also see it in solution. And so we decided to publish this. Of course, as you can imagine, we had a lot of trouble. We were kicked down the road from the best journals. And finally, we got some kind of a red carpet treatment so have I finished 30 minutes or 25?
Yeah. 2 minutes. 2 more minutes.
To speak and then for questions. Okay, great. So very good. I've conveyed the main message. So we got a red carpet treatment in a hoary old traditional journal called Archives of Biochemistry and Biophysics. And we described both that this was a novel fluorescence and that proteins should conduct electricity. And some years later, I was informed that there was a paper in the literature which actually confirmed both of these. And they used essentially the same explanation that we had used about partial delocalization of peptide electrons involving hydrogen bonds and so on. And they actually stuck some conductive AFM probes across an amyloid fiber made with So in our paper, we had described several amyloids, but this was a specific amyloid made with a synthetic peptide. And they made voltage current measurements showing that, that both of our observations were true. And they had somehow missed citing our work in their first paper, but that was corrected later. So it all turned out very well. And we incorporated that into a second paper. Then there was an invention. And then we got interested in the phenomenon and did a lot of further work looking at the excitation peak, which is 387 nanometers, emission peak in one particular instance, lifetimes we measured for this fluorescence, showed that there is a conformation dependence in the crystallines, where we know already that there is conformation dependence. We also showed that there's conformation dependence in lysozyme. I'll only say one thing here, that the fluorescence persists in unfolded monomers. And that has great potents for IDPs and amyloids and so on, and perspectives to consider for kinetic partitioning of molecules into, from IDPs into amyloids and so on. And this we think might happen because of hydrogen bonding with water. And so there is also incidentally a circular dichromosome band associated with this. I'll go straight to the last slide, having said that many groups now have now confirmed these observations, but I'll go straight to just one. I actually had, I think, 3 epilogues, but I'll talk about this. This is the work of Ubersky and colleagues. They have actually shown that individual amino acids also show this fluorescence. And what we think is happening is that instead of the lone pair from here jumping, electrons here quantum tunneling through hydrogen bonds to other peptide bonds, they can do that to water and to a cluster of structured water involving hydrogen bonds. And that is what allows a dispersed fluor to exist here as well, and allows them to see some kind of a form of the very same fluorescence now from amino acids. But interestingly, they don't see it from proline. And those of you who know the side chain of proline would immediately understand why proline does not show this. It's a very low quantum yield and very low extinction coefficient fluorescence in amino acids. But essentially, the same thing now gets put on steroids when it goes into a peptide. Thank you.
Okay, well, it's open for questions. Yeah, Daniel. Yeah.
Oh, you have.
No.
For it now, as you mentioned, other people have shown it too. I think the Kaminsky group have shown that you need amyloid. I mean, in their case, the fluorescence they see is specific for amyloid. So, do you have any comments to that? Is that because in their case it's another kind of fluorescence time resolved, and they need a?
I'm glad you asked that question. Actually, it's seen in amyloids, and we had reported that in the 2004 paper. But it's seen in every other kind of protein form as well. But in amyloids, it actually is very striking because you have all these protein molecules organized into cross-beta sheet structures. So the rivers of electrons running through are really good. And that's why you have those voltage current curves, which were there in that PNA's paper from Italy. So it is most striking in amyloids, allowing amyloids to act as conducting fibers for biomolecular electronics. But it's not that it is seen only in amyloids. It's seen in every kind of aggregate, including amorphous aggregates, crystals, powders, and proteins in solution.
Okay.
So,
thanks.
Yeah.
Those last spectra where we measured lifetimes, those were with 100 milligrams per ml of lysozyme and gamma crystalline.
Yeah.
Hi.
And then back there.
Nice talk. I have a question in the sense. So from the fluorescence emission spectra of these amyloids or whatever fluorescence you described, can we get the structural information from intensity of different bands at
by
wavelengths?
deconvolution, I think we will, but I think we await some more refined instrumentation. Also, I think today morning there was a talk about internal hydrogen bonding in intrinsically disordered proteins that maintains some kind of backbone protection from the solvent. So I think that in intrinsically disordered proteins, we are going to see a fifth kind of form of this very same fluorescence. In unfolded proteins, as you can see, with 6 molar guanidinium hydrochloride in lysozyme, you see the fluorescence is not just It didn't go away, it intensified, and it changed in character. So it'll be very interesting if all the people who are working with IDPs look for this fluorescence and use it, because it's a global probe for the whole protein, because there are peptide bonds everywhere.
So we didn't see it in tau by itself, but we saw it in tau everything.
Yes, so I think the issue there is one of quantum yield, basically. Yeah, so the quantum yield is dependent on the structure, and so it might be very much higher for amyloids, like Daniel is saying, than it is for others. But if you look hard enough, in fact, I would say that if we had looked hard enough with amino acids, at that point it didn't strike us that it could be amino acids. We just jumped straight to the possibility that it's peptides. We would have probably done studies with high sensitivity and seen what Uversky and Muschol have now seen, which is that the carbonyl group itself can also give rise to this fluorescence, presumably by allowing those electrons to jump into water, and then from water to water to water, and so on. So, but that'll be much lower quantum yield. So if you don't see it, and you see, we didn't see it in glutathione. And that was good because it helped us to frame the hypothesis. If we had seen it in glutathione, we would have been stumped. But now in retrospect, if we go back to glutathione and do a very high sensitivity study, I'm sure we'll see it in glutathione as well. After all, if you see it in amino acids from the carbonyl moiety, why wouldn't you see it in glutathione? But it's much lower quantum yield than a peptide which is hydrogen bonded to other peptides in hydrated powders.
Okay, yeah. Please.
So, the transfer of electron for conducting the electric signal. So, the protein need not be bound by covalent bonds. I mean, is jumping just like, let's say, from one peptide to other peptide?
Yes. So within a monomer, the electrons are not following the backbone. They are following the network of alternating hydrogen bonds and peptide bonds, right? And once they do that, so, of course, they are following the line of a helix because that's how the hydrogen bonds are organized. But in a sheet, they're going transverse. But once you cross over from one monomer to another monomer, which is in contact, one presumes that if there are edge-on hydrogen bonds, that's just an extension of the protein into another protein molecule, and so the process can continue. So in all of these electron transfer systems in the mitochondria and in chloroplasts and so on, one presumes that this would happen. It would also allow different embedded organic molecules within these protein matrices to be in quantum coherence with each other, to talk to each other through these conducting, through the conducting matrix in which they are embedded. So you can have both a knowledge of the state of one known by another organic moiety, and you can also have electron transfer, which is virtually instantaneous. Because you push in an electron here, an electron leaves from the other end of the river.
Yeah, well, the time is. I have one quick question. Have you considered to be phosphorescence instead of fluorescence?
Yes. So phosphorescence would, of course, involve much longer gaps between the excitation and the emission. And the 3 lifetimes that we measured, which have now been also confirmed by Kaminsky and Dobson and by others, including Uversky and Muschol, they are basically, one of them is in a few nanoseconds, and the other 2 are sub-nanosecond. So that clearly shows that they are fluorescence and not phosphorescence.
But it's strange because on the other hand, the quantum yield is very low. But yeah, but it's very interesting, yeah. Well, let's thank again our speaker.